Today we have two speakers joining us. Dr. Pete Rand is with the Prince William Sound Science Center, and then Dr. KRISTEN Gorman with UAF. And they are going to talk to us today about the Copper River Salmon 2125 program. Pete, go ahead.
Great.
And here's right there.
This is going to work. Okay. Yeah. Great. Okay. Welcome. Yeah, I'm Pete Rand. I'm at the Prince William Sound Science Center. Started here as a staff scientist back in 2015. I've made some presentations as part of this lecture series in the past. And this is an important week for us. So we're holding a workshop. We're calling it Copper River Salmon 2125. Started this last year. So we're thinking basically long term. We're not really focusing on kind of short-term changes that you might see in salmon. We're taking a longer-term perspective, and we're convening a lot of folks together in Cordova this week. So we've just spent the day with about 30 folks from both up and down river, and we covered a lot of ground already. It's a 2-day workshop, so we're trying to pull together a lot of information about the status of Copper River salmon, and again, taking this longer term perspective, trying to understand how climate change might be impacting these fish. So KRISTEN and I are going to kind of just briefly describe some of the trends that we're seeing in the Copper River, and we're going to focus on some studies that we've been collaborating on in recent years. And we'll probably be touching on some of the things that we learned today. We just had a series of 30 short presentations today, and so there's a lot to absorb. It's important to emphasize that we're kind of focusing on the copper, but we're bringing in examples, case studies from elsewhere. We heard from Scott Hinch from the University of British Columbia, and so he provided a lot of important information on what they're seeing in British Columbia and what we might be seeing in Alaska in the coming years. So anyway, this is what I want to cover. So I talked a little bit about our workshop, and we're emphasizing that we're carrying on this tradition of convening. I know RJ Kopchak, who's a co-founder of the Science Center, started a lot of this work on building a Copper River knowledge system, and then the Copper River Watershed Project, and others have held a lot of these workshops over the years. So this is another example of importance of communicating among research specialists, but also among stakeholders, bringing a lot of this information together and sharing it. I'm going to spend a little bit of time just talking about trends, specifically environmental conditions and how they're changing, both in the ocean and in fresh water. Of course, salmon are anadromous. They spend part of their life in freshwater and part in marine. So I want to talk a little bit about that. And then KRISTEN and I have been focusing on studies of the spawning migration period for sockeye. So we're using a number of different tools, radio telemetry, looking at body composition, energy, and we're looking at pathogen load and diversity, and trying to get a kind of a broader picture on salmon health. And I should emphasize this is just one part of a much larger group of research folks studying a lot of different things in the copper these days. So we're going to be focusing on our work there. And then an important deliverable from this workshop is to try to work toward a life cycle model. So we've been focusing a lot on the adult migration phase, but there's important effects going on at different life stages. So we want to try to broaden this to look at the full life cycle. And then I'm going to, at the end, just talk a little bit about our workshop and where we're going. This is a map that RJ provided, RJ Kopchak, and so he and folks at EcoTrust, based in Portland, did a lot of this early mapping work, raised a lot of awareness about the size and scale of the Copper River and its importance, and importance of glaciers, and importance of native communities and the commercial fishing operating on the flats. And so I know you probably know a lot about this, but this is a really important watershed, of course, and glaciers, of course, are a really important part of this. So I'm going to be focusing on the importance of glaciers in terms of temperature and discharge in the river. This is just a couple different photos. This is RJ right here. Again, he held a series of workshops. There's actually proceedings from those workshops that were produced. And then this was our last workshop in science synthesis effort. This was done at the Taslina Hall, village of Taslina in the Upper Copper. This was held right in the early part of the pandemic in early 2020. So we really haven't had an opportunity to convene since then, but we've been able to do a lot of work in the intervening years. And so part of this workshop is sharing all of the great work that's been done since this meeting. But this was really, this meeting inspired a lot of new work. This came after 2019, which is the heat wave year. We'll both touch on that year. But there were a lot of reports from users that the salmon were in pretty rough shape in 2019. And this really got us thinking about salmon health and understanding salmon status better in the copper. And then this is a picture from last February. This is the meeting that we held in Copper Center. Karen Linnell is the executive director of ATRIC. This is Matt Pichet. Matt is with the native village of Eac, and he was describing some of the work that he's been doing in the copper. But again, this is the kind of start of our workshop series. So this is the second in a series of workshops, and I just want to acknowledge the National Park Service, Wrangell St. Elias, for providing support for these workshops. Okay, so this was highlighted in a number of talks today. This probably is not new news to any of you, but marine heat waves, particularly in 2015 and 2016, had a kind of systemic effect on the Gulf of Alaska. It was not just the surface waters; it was warm water down deep during those years, and we saw lots of changes. So this is anomalously warm temperatures shown here in red during those years. And the idea is that these events are becoming more frequent, and they're affecting salmon, the survival rate and the growth rate. And so it's important to recognize that. We also are seeing competitive interactions with other species, including pink salmon, wild and hatchery pink salmon. So the real key part of understanding salmon status is understanding how oceans are changing. We're also seeing trends in river temperature. This is data from Fish and Game. This is water temperature in Celsius over a number of years. And these are temperature loggers on the north bank and the south bank. And so these are for each season. They basically, Fish and Game puts these loggers in at the beginning of the sonar season, takes them out at the end. It's just important to recognize the warming trend in both of these time series. Of course, the north bank is a little bit warmer. The south bank is reflecting more flow out of Miles Lake, which is influenced by a glacier, so it's slightly cooler. But important to just recognize that there's a long-term trend of increasing temperatures. These are still relatively low temperatures. We're talking about stress on salmon. We're talking about something, temperatures much higher than that, 18 or higher. And so I should mention, Scott Hinch basically presented very similar trends in the Fraser, but of course, as you would expect, we're much warmer. So they're getting 18, 19, 20 degrees in the lower Fraser. So, yes.
Stop you. I don't think we're showing up online. I just want to make sure. Give me 1 second. Okay, there's our camera. Can you guys online see the screen?
I can see the screen. I can't see the presenter. But
Okay.
you just turned the camera on for the presenter.
Okay, you can see us now?
Yep.
Okay, sorry about that.
Both the slides and the presenter. Well, whoever's speaking.
Thank you so much. Sorry about that.
So, yeah, these are the temperature time series from the lower river at Million Dollar Bridge. And then if we go upriver, this again is Alaska Department of Fish and Game data. This is at the Gulkana Tower. So this is several hundred kilometers upriver. And this is a river system that has very little glacier influence. So it's a relatively warm water system in an otherwise glacier dominated watershed. And these are different years. So 2016 to 2021. And then this is that warm year that I mentioned, 2019. And so this is the 18-degree threshold above which we think these fish are becoming stressed and more susceptible to infection and mortality. And so you'll see, especially in 2019, a number of consecutive days where the temperature was higher than this 18-degree threshold. So so I think at least in the Gulkana, we're seeing temperatures warm enough to significantly stress fish, salmon. And that's temperature. So we're also seeing changes in flow. This is USGS gauge data. This is discharge in cubic meters per second over the course of the record here. And again, 2019 was a really high discharge. So in most all other salmon rivers, when you get these heat waves, you see drought conditions and low flow. That's a very different situation in the copper. You get these really high discharge events. And then this is Melissa Valentin developed a model. She actually presented some of this at a science symposium here in Cordova a number of years ago. And it was really, to me, a very eye-opening presentation because it projected, based on downscale climate data, what discharge will look like in the copper on the next 100 years. And so that's this model output in blue. So this is projected over the next 100 years discharge based on what we expect the rate of glacial melting to be in the system. These are this is an earlier publication looking at mass balance. Mike Loso with the Park Service has a new study looking at this in more detail. But the basic message is that glacier melting is increasing, and that's increasing the flow. I should mention this is during July. This is where this is significant because this is when most of the fish are swimming upriver. And so the idea is they're getting faced with higher levels of discharge. This we think is really important in the Wood Canyon, which we'll touch on later in the presentation. And then what really kind of started all this discussion is this concern about declining size. This is, again, fish and game data going back to the mid-60s. This is for a 5-year-old returning sockeye salmon. And so in recent years, well, in 2014, 15, 16, we got unusually low size small fish returning. It has rebounded somewhat in recent years. Jenny Morella with Fish and Game presented some more updated data on this, but it's still very low compared to the historic average. So one of the kind of central questions with our work is, what does this mean in terms of migration success? These fish are facing usually high discharges and in some cases warm temperatures in the upriver. How might this be affecting their migration success? And then there's another recent study that I think really helps illuminate what climate change might mean for salmon, particularly in the Copper River. This is work done by Kara Pittman. This was published a few years ago, and it got a lot of attention. It's an excellent paper, and it's looking at as glaciers recede, there's new habitat that's forming, right? And so this is this could potentially boost production by unlocking habitat, creating new spawning grounds. And she just presented today on her work on lakes, proglacial lakes. So with satellite data, she's able to measure temperature of these lakes, and as they become more disconnected from glaciers, they clear and they warm up and they become potential new habitat for sockeye in particular, for rearing fry. So this was her earlier work, and basically the take-home message is that the copper in parts of the northern Gulf of Alaska might see a boost in, very likely we'll see a boost in production, and just in terms of numbers of kilometers of river accessible to anadromous salmon. And so she estimated 1,000, basically 1,000 new kilometers of streams in 100 years. So at the end of this talk, I'm going to incorporate this kind of dynamic in the model to try to understand what that might mean in terms of overall abundance and run strength in the Copper River. I should note that, of course, yeah, there's going to be the story is there's going to be no new production basically in the southern range because of a lack of glaciers in that part of the salmon range. And then finally, I think many of you are aware of this. This again in 2019, we saw many cases across the state. All the species of Pacific salmon found in Alaska. We found numerous cases of premature mortality. So this was a picture taken with one of our field crews in Prince William Sound, and it again affected many fish, primarily due to warm temperatures and low dissolved oxygen. We saw many cases of mortality, and so this, I think, 2019 was an important year to really understand what the future might look like for salmon. It should be noted though, the fish came back very strong in 2021. So even though it would look dramatic, I think that, yeah, the salmon can be quite resilient, and the run strength from this brood year was actually quite strong. So they managed to successfully spawn, even though we saw many cases of pre-spawn mortality that year. Okay, so we're gonna shift gears a little bit. KRISTEN's gonna talk a little bit about some of the work we've focused on in this 2019 to 2021 field project.
Okay, thank you. This would be, okay, great. Hey everyone, I'm KRISTEN Gorman. I'm up at UAF in the Marine Biology Department, and I started working on these issues with copper river salmon with Pete when I was here at Prince William Sound Science Center. And I gotta say, it is very satisfying to see a lot of this start to come together. So thanks for the opportunity to talk to you all today. Pete and I have worked pretty hard to get some field studies started. Basically, we began in 2019, and this kind of initiated with funding from North Pacific Research Board after the 2018, it was a bad run in 2018, right? Or I'm sorry, yeah, it was, I think. And so you can imagine kind of this like reactive nature of science, right? We have a bad event and then we got funding, right, to do this work. So we were able to piece together funding from NPRB as well as Alaska Sustainable Salmon Fund. And so that allowed us to conduct this 3-year field campaign looking at kind of quality and health predictors of migration success in the Copper River, specifically for sockeye. And so our projects over these 3 years kind of had 2 components. One was to conduct a body composition analysis where we really wanted to quantify aspects of body size and energy content and reproductive investment and how that varied across different stocks that were returning either very far in the river or kind of at mid parts of the river or lower down in the upper Copper River. That lethal sampling has been really informative because it actually gave us information on reproductive investment. So things like fecundity and egg size. And we sort of are applying that framework to our results from a radio tracking study. So we also radio tracked fish over these same 3 years. That's this image here. So Pete did a lot of the radio tracking study. We worked with collaborators at NVE and basically captured fish in their fish wheels lower down in the river, tagged them, measured size, mass, energy content, and also gill tissue samples for some molecular metrics of stress and physiology. And what we're really trying to do is improve our understanding of migration success and the factors that affect that. And I will mention, if anyone's interested in a more in-depth, Abby Host is a grad student on the project, and she's going to be defending her master's thesis this Friday. And she worked up the body comp data for this project, which I'm going to talk about here. Yeah, so sorry, this is just radio tagged fish. This is our microwave fat meter that we ran along the outside of the fish. This gave us fat estimates in a non-lethal manner that we could apply in the radio study. And then this is just an image of us doing some of the lethal collecting, I guess, upriver, and probably grabbing gill tissue samples there. So we had great crew, and it's just been a really fun project. Yeah, this is just a picture of the fish wheel. And again, working with NVE has been just instrumental and critical to pulling this work off. And this is really what allowed us to collect fish in good shape that we could radio tag and then send them on their way to try to understand their migratory journey. So this is just a results graph, and this is actually some of Abby Host's master's work. And so this is related to the body composition study. So in this study, so over the 3 years, we actually collected fish in the lower parts of the river. We collected about, what was it, 20 fish per run timing group, so about 60 fish each year. And then we compared aspects of body size and energy content and reproductive investments in the lower river with fish that were successfully reaching their spawning ground sites, so these successful migrants in the upper river. And what we've done is at a stock level, basically calculated a metric of migratory work. So this is essentially distance in the watershed and the elevation that a given population is returning to. And what we're finding is these sorts of relationships where, interestingly enough, there was not a strong relationship with body size and migratory work. There's past work in the Fraser River that suggests, you know, these quality metrics of fish are highly shaped for their migratory journey. And we actually didn't see a relationship with body size and where they end up in the river. What is interesting, though, is that we have this fairly strong relationship between kind of the ultimate destiny of a fish in the river and what their somatic energy density is as they enter the river. So this top panel is for fish that we collected from the lower river. The bottom panel is for fish on the spawning grounds. And then, yeah, we had another kind of energetic metric, which is total somatic energy. It's essentially energy density multiplied by the weight of the fish. And we actually didn't see a strong relationship in the lower river for total somatic energy with migratory work. What has been interesting to us is how these results compare with what we have measured on fish on the spawning ground. So we can kind of think of these fish as successful migrants, right? They actually made it through their migratory journey up to their spawning grounds. And one of the things we see is that these relationships, so the R-squared values, increase quite a bit for fish on the spawning grounds. So again, really strong relationships between energy density and how far they're reaching in the watershed for their spawning ground sites. And what was also interesting was this really strong relationship that came out from total somatic energy. And the way I think we're interpreting this is that perhaps body size and certainly energy seems to be a strong predictor of whether or not they're actually making it to the spawning grounds, is I think how we're thinking about it. So I guess I'll, leave that there. Another thing we've been looking at is essentially health of these fish through the lens of pathogens and also molecular transcripts. And we basically did this work with collaborators at the Department of Fisheries and Oceans Canada. They kind of pioneered a lot of these techniques with the Fraser River sockeye system. And we've been really lucky to kind of learn from their experience and apply this framework to the copper. So during our studies in 2019 and 2021, we collected gill tissue samples, and we're basically using this fluidigm array to do real-time qPCR work that's really detecting the loads and presence of pathogens. And I guess I want to be clear that this does, if we detect a pathogen in a fish, it doesn't mean that that fish is diseased. It's not an estimate of clinical disease, but it gives us a sense of what kind of pathogens are out there, which is really new information for the copper. So it really allows us to identify microbes in wild salmon, and we can kind of restrict our surveys to microbes that are known to be causative of disease. And it gives us a little bit of a window into potential disease ecology in the system. And so I guess what I would say here, these are data from our collected body composition fish in 2016, our initial pilot study in 2019. And some of the things we've learned from these data, and so these are the 2016 fish, and this is just prevalence of all of our given pathogens. So there were over 40 that we surveyed for. They include bacteria, parasites, and viruses. And, you know, one of the obvious things is that in 2019, that was a fairly warm, that was our warm year, right? We started detecting new pathogens that we hadn't previously. So we know that temperature is highly associated with pathogen and disease dynamics. So, you know, perhaps under warming temperatures, you know, we've got the presence of new pathogens that are showing up. We are doing some work looking at kind of the energy density of sockeye as they enter the copper. And so these bar graphs here are showing the energy density of fish sockeye that we collected in the lower copper and at Miles Lake, sort of lower in the river, and then on spawning grounds, in this case, the East Fork, Gulkana, and Mentasta Lake. So you can see they're using quite a bit of energy to reach their spawning grounds. And from a pathogen perspective, fish with higher energy density typically have lower pathogen richness, so a lower number of pathogens that we detected in their gills. Once they get on spawning grounds and have completed their journey, we're kind of seeing that they have higher pathogen richness, which might suggest that maybe they're picking up some of these pathogens in the system. And we're going to, you know, with our studies, learn more about what that means for quality of the fish and what some of these relationships are. So this was, yeah, I guess kind of a summary of our body comp study, and then we can kind of use that in the context of how we're thinking about the radio telemetry work. And so basically with the radio telemetry study, we're actually able to understand the fate of these fish and what the predictors are of their quality in the lower river and how that plays a role in their fate. Our body composition data suggests there might be important roles for body size and energy density, and that's actually what we're seeing from these analyses. One of the things to note is that fat content was increased for fish that were successful, and also it looks like, yeah, I guess, sorry, what, this is discharge?
Discharge, yeah.
Or,
Survival.
oh, on survival, yeah, okay.
Yeah.
So reduced survival with discharge. So I think, yeah, we're kind of starting to paint these pictures from a quality perspective on what the ultimate fate will be for these fish. And we're still doing a lot of work with this data set to pull in both the transcriptome analysis and pathogen diversity and how that is related to fate of the fish system. So, that it?
Yeah, then I can pick that up.
Yeah. So Pete's going to talk about how we pull this all together in next steps.
Great. All right. Thank you. Just, yeah, a few more slides to give you a sense of where we're going with this work. So we learned a lot through that 3-year study. We learned quite a bit. And again, that 2019 year, I think, was really got us thinking about what kind of conditions lead to in-river mortality of these fish. And so, yeah, this was this is something Lizzie Turner, who's been working with me for a while now, plotted all this information. So these are the fates of the fish during that year. It wasn't a great year because we got our tags late, so we missed the kind of early part of the run, but we were able to tag the mid and late season run. And all of these red tags are tags that are motionless, basically indicate that the fish died. So it's a mortality code that's emitted by the radio tag. So when you fly over the river, you can detect where all of our tagged fish are and whether they're alive or dead. And this is a compilation of all the aerial detections. And you'll see, yeah, most of these fish were dead. We were tagging them here in the lower river, and most of them never got through Wood Canyon. And this is very unusual. We've done a number of years of telemetry after this, and then Native Village VIAC has done early telemetry, and we never saw a pattern like this before. There's some question about our capture methods, but even the fish that we caught in the fish wheel basically showed this pattern. We were also dip netting fish in side channels. These are fish that probably were in relatively poor condition. But it was, yeah, basically clear from that year that there's certain conditions that elevate in-river mortality. And the great part about our telemetry project is that we were able to measure all of these characteristics on the fish before we released them. And so we looked at the fates of all of the fish, looked at sex, males and females, fat content and discharge, as KRISTEN mentioned. And these were significant factors. So females are vulnerable. They're more susceptible to in-river mortality. And this is supported by a lot of the work being done in British Columbia as well. And that could lead to skewed sex ratios. I should mention that the volcano hatchery is now seeing a very skewed sex ratio. So they're up to like 90% of the fish coming back to crosswind are male. So we think that's that could be a result of this sex bias in survival. And then, of course, as you might expect, the fish that are in better condition have higher fat content do well, and fish that are faced with high levels of discharge have poorer survival. So we had these 3 years of data. So we wanted to pull this together and try to understand what it might mean at the population level, and kind of understanding how the fish might cope with future conditions. So through this workshop series, we're working on building a model to try to capture some of these dynamics. And why are we doing this? We're trying to look at how changing river and ocean conditions I described might affect the fish and then fisheries and livelihoods of folks working in the Copper River. So understanding this from a sustainability point of view, we want to track these population dynamics and try to understand what ecological factors may be important. Climate vulnerabilities. So there's many different populations. They all, as KRISTEN mentioned, they all have different levels of distance that they travel to the spawning grounds and level of difficulty of that migration. So we want to look at climate vulnerabilities and what populations might be more sensitive to change. And then we want to look at this idea of resiliency. And so, as I mentioned, copper is very unique salmon river system because of the presence of so many glaciers. And this could potentially provide a buffering for warming temperatures and also boost productivity by, again, creating this new habitat as glaciers recede. So we want to evaluate all of these things in the context of a model. This is something I presented today just to emphasize that salmon have very complex life cycles. And we have to make a lot of simplifying assumptions to try to track population dynamics. And again, we're focusing for this project on reds or sockeye salmon, but a lot of these same things are occurring in other species, particularly Chinook. So even though we're focusing on sockeye, I think there's a lot of interest in extending it to Chinook. Okay, so this was an early paper done by Mike Healy. He was my postdoc advisor at University of British Columbia. And so he was one of the first to really understand cumulative impacts of climate change through the whole life cycle, from eggs all the way to returning adults, spawning adults. And he published this in the Canadian Journal a number of years ago. And I recently reread it. It's definitely an important read. And I've actually contacted him in recent years, and he still stands behind this. It's kind of sobering to read through, but he walked through every step of the life cycle and tried to understand, at least at a qualitative level, how climate change might impact salmon. So he looked at freshwater conditions, scouring of the reds, early embryo development, the phenology or timing of migration and life history phases, looking at the warming ocean and how that might affect conditions, and then of course flows and temperature in river, And so, and this was again based on a lot of work in the Fraser. So this got me thinking about the value of kind of moving from a qualitative model to a more quantitative model. So this was a kind of a simple diagram that I presented in an early workshop, just emphasizing the fates of the fish returning to the river, caught in the flats in the commercial, the Copper River commercial by the fleet in the Copper River. There's subsistence catches, dip nets, and fish wheels in the upper river. There, as you saw in that 2019 data, there's some growing concern now about in-river en route mortality. There's egg take at the Gulcana hatchery, and there's been concern about getting enough brood stock in recent years. And again, that skewed sex ratio is a concern for hatchery managers there. And then, of course, ultimately you want fish on the spawning ground. So this is the framework, the kind of modeling framework that we presented at the workshop last year in Copper Center. And we're going to be focusing a bit more on this tomorrow at our workshop. But again, it's a little bit more detailed representation of the spawning migration, emphasizing these different stages when fish returning from the ocean, swimming through the delta, getting counted at Miles Lake, and then going into different drainages in the upper Copper. And at each stage, we have representation of harvest, commercial harvest, and then personal use, subsistence, and sport fish harvest in the upper river. And I guess the important thing to see here is that there's controls here in the life cycle that's related to temperatures and flows. So our interest is to try to relate this migratory success and survival to these conditions, and ultimately make this a life cycle model where all the spawners basically feed back through a stock recruitment relationship to fry and smolts re-entering the ocean. So this is the kind of framework we're focusing on. I wanted to just give you output from kind of a first stage of this model effort. And really what I wanted to look at was how what the net effect might look like if you see these glaciers receding, creating new spawning habitat, but also seeing these higher risk of elevated in-river mortality from fish coming back to the river. So a potential positive effect, a boost to the population, but also a growing threat in terms of higher flows in the lower river. So this is again Kara Pittman's work with a kind of more detailed diagram here showing is that glaciers recede, you're kind of creating new habitat here. And again, she had predictions on how many kilometers of river will be created through this glacier recession dynamic. So this is just a projection from the model. This is kind of the baseline through the early 2000s here. And the red basically is indicating the numbers of fish lost from higher discharge, this in-river mortality occurring from high discharge. And so you see a very clear spike here. This is that year with very high flows, 2019. And then as you project forward, this is again Melissa Valentin's discharge predictions based on climate change. You see this mortality risk increasing over time, with some very dramatic cases where fish are blocked and suffer high mortality. And at the same time, we're seeing this green line is indicating the amount of production that we might see as these new habitats as they're being created as glaciers recede. And so this is all in the kind of same units. This is numbers of returning fish to the spawning grounds. And so I guess one of the take-home messages is that there's a kind of growing risk we see in terms of higher flows in the lower river. This is a characteristic of a system that's dominated by glaciers. But we're also seeing this potential boost in production. This is just looking at creation of these new streams. As I mentioned, Kara talking about how this warming trend might enhance production in some of these nursery lake systems. So that's what they're actually seeing that in Bristol Bay with warming trends. You're seeing more production of fry in some of the Bristol Bay lakes. So anyway, this is kind of a first cut at sort of understanding what it might look like in 100 years' time. Just looking at these two dynamics that salmon are going to have to face in the future. So yeah, I just want to conclude. Again, we're meeting these next few days. We just finished up our first day, and we're really trying to understand, continuing to understand who's doing what where. And so we're kind of describing the salmon data landscape. And we're getting a lot of support from ATRIC. Casey Cusick has been developing this online mapper where we're trying to get a lot of this information online so it would be easy to access and share information through the internet. And we've got lots of people sharing knowledge, observations, and perspectives. We've had a lot of that today. We'll continue that tomorrow. And a lot of what we're doing is focusing on what are the key metrics and indicators. So through our work, my work with KRISTEN, we've kind of developed approaches that have been used in other systems, but we're applying for the first time in the copper. So, but there's lots of different perspectives and ideas for that. So we're going to be focusing a lot on understanding what we know and what we don't know, and trying to fill in gaps in our understanding. And I guess another thing that makes this group unique is that we are really taking this long-term perspective. We're looking at it 100 years into the future. And I just want to, emphasize that I think this is a really important approach we're taking, trying to understand the whole life cycle of salmon and focusing on these different life stages. And then, in terms of outcomes, we're going to be talking about next steps and what we might do. But we're really, I think, encouraged by a lot of the new work being done by a number of different players in the copper. It's really kind of gratifying to see that. So that's all I've got. So I'm happy to take any questions if we have time. Looks like we have some time. Thank you for your attention.
So Pete, you mentioned that 2019 year return several times.
Yeah.
That brood year has come back now. How was the return from that brood year like?
Yeah, can anyone, is anyone here from Fish and Game?
Don't look, Rachel.
Rachel?
I'm not gonna speak to that offhand. I'm not sure off the top of my head. Yeah.
Yeah, it's definitely worth looking at. Yeah. Yes.
Yeah, for the fishermen, I can tell you that 2024 was a pretty good season on the copper, just anecdotally. But what I really wanted to say was I really appreciate your and KRISTEN's research in the river, looking at how important it seems to be for fish mass and body energy density. That it seems to beg the question of ocean research.
Yeah.
What's going on out in the ocean?
Right.
They seem to not be getting enough food. And is it mainly because there's simply not the food there? They have to travel farther and work harder to get it?
Right.
Or are there predation things going on, competition with other species or with other salmon species maybe? And so that would be wonderful if people could really start throwing some money at ocean studies.
Right.
And get at some of those issues. It just seems so key to this whole thing.
Yeah. Yeah, absolutely. We had a special section today on ocean. There's folks doing trawl surveys and looking at juvenile stages along the coast, and they're making a lot of the same measurements that we're making on adults. So they're looking at condition and size and species mix. There was an International Year of the Salmon that happened. There was a lot of focus internationally on understanding distribution of salmon in the ocean. And clearly, yeah, the surface temperatures are getting warm. That increases the metabolic demand of the fish. So, and then there's been relationships between prey, plankton densities, and growth rates. And then, KRISTEN's done some work looking at isotopes to try to understand, carbon sources with what they might be feeding on and whether they might be shifting the different prey during that stage. So it's definitely an important thing. It's just a lot. Yeah, there's a bigger price tag to work out there. But I know, NOAA and Fish and Game basically Fish and Game specifically have started a new ocean ecology program. So they're focusing a lot more on ocean conditions and understanding how they're impacting salmon.
Yeah, I feel like that is like one of the key data gaps that we should talk about tomorrow in the workshop. And one of the things that struck me today was from Wes's talk. I mean, it seems like they've had pretty consistent sampling in Southeast by NOAA and then into the Bering, but the whole central western Goa.
Yeah.
I mean, other than the IRP that went on has been like, I don't know why that's left out. I was gonna ask him that question.
Right, yeah. The whole Copper River plume and that part
Yeah.
of the Gulf, there's been no long-term efforts there. It'd be really fascinating I know we've had a lot of internal discussions about the importance of doing work closer to home.
I would mention, sorry, one more thing. There are other types of data sets. So out at Middleton Island, there's a bird research station, and they get a lot of little small fish samples from the birds feeding their young. And so there's, things we can tap into, but it certainly doesn't take the place of like an actual sampling program.
Rachel?
I have one comment too, just talking about estimating success in the food year. To estimate total run size, that's both escapement as well as catch information, right? That means that total run estimate for a particular age class. And so right now in the Copper River, those catch numbers are not apportioned by Upper Copper River and Lower Copper River stocks. And because of that, it can be hard to kind of give an estimate of the success of any particular food year in the Copper. It's actually working on a project that's looking into, trying to better resolve that right now. But yeah.
Yeah, there's new work on genetic stock identification too, which is really right. Exactly. Yeah.
Can I ask a question too?
Sure.
Maybe I missed it, but the data that was input into that model projecting in river mortality across that time series.
Yeah.
What was that?
That was the risk. So we developed a relationship during those telemetry years.
Okay.
And we related that to discharge level in July, with the discharge that they're facing during the adult migration. And then we use the projected discharge from Melissa's model to estimate, the mortality risk under those conditions in the future.
Okay, so from those 3 years.
From those 3 years, right? Yeah. Yeah.
Sorry, Rachel, can I ask one? So when you mentioned total run size, escapement, and harvest, right? But there's also natural mortality that goes on that we probably don't know. I mean, it's hard to quantify that, right? But
yeah,
I think that's one of the things that our telemetry work is really at least hammering home for me, is that there are some fish that are just not successful that, yes, they got through Miles Lake and we think that's escapement, but they died, they didn't make it, so.
Yeah, and it's important. Scott mentioned this in the case of British Columbia, they're incorporating that information. So they know the conditions upriver, and they know that under certain conditions, those fish are going to suffer high in river mortality. So they're incorporating that in the management. They're much further along just in terms of understanding how these river conditions affect mortality risk. But I think these kind of telemetry studies, I think, can help inform that, especially, I think this discharge effect in Wood Canyon is going to be really important to keep a better eye on. Yeah, Jim?
Yeah, and sort of related to that whole in-river mortality, you know, at least Pete, that this has been a drum I've been beating for a while. And what some of the other department staff have told me is that basically the assumption right now is that the in-river mortality is the same from year to year. That it is incorporated into the models and the schemical, development, etc. But now we're learning of this interannual variability in that in-river mortality, and that's what has been taking place with some of the research projects, with ichthyophonus in particular on the Yukon, in order then to evaluate some of how they would plug that in to an in-season management tool and a predictive aspect. And one of the challenges was, though, that to do that particular work in the Yukon, it required sacrificing the fish, and yet we have a moratorium and all of that. So even the tribes and First Nations were opposed to the sacrificing of those fish because it's so dire now. And so that's the importance of trying to get ahead of it now, right? And to not wait until, like you said, 2019, then you got reactive research, right? We need to be proactive and get ahead of the game here. We know from even though the Yukon River is a very different system, we know that in-river mortality isn't necessarily constant from year to year, right? And nor will it be in the future if changes continue to be dramatic.
Yeah, right.
So let's put heads together and figure it out, right?
Yeah, Matt.
The somatic energy density figure that Kristen presented, I think the chart with the strong relationship, there was an X way up on the top. Is that the Galcana? And
Yeah.
You should focus on that. This is
What
another
do you think
interesting story. Yeah.
Yeah,
And was that sample?
good eye.
And was that sample? I mean, did you see like sex ratio bias there or anything?
That I don't know. So yeah, there are, let's see, so there's this X, this is the Galcana hatchery. So we did not include it in our regression analysis because it's a hatchery stock. And then, so that's, what is this? This is the lower river, right? This is the upper river. And then for the upriver, we also didn't include Long Lake because they have a curious life history where they spawn very late. So they're actually have a relatively low, where is Long Lake? I think this is Long Lake. A relatively low migratory difficulty, but very high energy because they spawn so late in the season. So we're trying to look at, how does natural selection operate in the system? We sort of left out the oddball populations. We didn't want to confuse that story. So that's what that's about. But one of the things that we are learning about the Galcana, which I hadn't appreciated after going through this exercise, was just they have like one of the highest migratory difficulties in the copper because of the elevation there. And they also return later. And so they encounter higher discharge. So yeah, I don't know. It's a curious situation. Eric.
Yeah, I guess getting back to Bill's question about if you added more about ocean conditions and how that affects productivity. I was reflecting, the two of you co-authored a paper a little while back with Brendan Connors, who was the lead, looking at climate and competition effects on sockeye productivity.
That's right.
And I was wondering if some of those broad scale relationships that you found in that study could be used to.
Yeah.
Right. Because you looked at populations way down south, where you're encountering a much warmer ocean that might be similar to what the Gulf of Alaska looks like in the future. Wonder if that might give some basis to make projections.
Yeah, that's what we're hoping to incorporate that kind of information. That was productivity. across, many populations. And we saw that, West Coast salmon were much more sensitive to sea surface temperature. And we saw a positive effect in the Bering Sea. In the Gulf of Alaska, the populations were intermediate. Yeah, we saw temperature and effects of that sea competition. So that would be something, be nice to incorporate that. Yeah.
Talk about sea temperatures. And there's years it seems like I can catch fish on my lead line. Don't catch them up on the cork line. So when you say sea temperatures, is that surface or such depth or, I mean, assuming it's colder depth, or isn't one temperature of the whole? So what's your standard temperature of?
Yeah, I know the blob that extended quite deep, right? Do you know?
Yeah, I don't,
The
have the-
common measurements at the surface.
Right.
There's not very many measurements at depth.
Yeah, and there's some telemetry data. They had temperature sensing tags, and the fish do this diel migration and can experience very different temperatures. So
Yeah, so I mean, sunny weather, if it's cloudy all summer, or, would it affect the surface temperature quite a bit? I mean,
Yeah. I mean, one of the things about the blob that we've learned is that the heat wasn't just at the surface. It went through the entire water column. I mean, not the entire, I think what,
yeah, it was
I
deep.
think
I don't know.
Down
It
to
was
200.
200 meters, yeah. And that's what, another feature of the blob that was pretty unique was just the depth at which the heat was being retained. Have a graph of it in the other talk, but.
I'm trying to find out there, okay, it was at depth, it was warmer, but how much warmer? What, was it a fixed number all the way down, or was it a ramp, I mean, it just seems awful good generality for.
yeah.
I don't know.
It was fairly fixed all the way down during the blob. I'm having to picture the
Copper salmon,
that Daniel's
This one?
graphs,
that one.
but it was red, kind of top to bottom. So it's like everything had increased, a degree or two Celsius. So,
Here's a Danielson graph, Scott.
Yeah, so,
but that's integrated.
Yeah, that's down to 250 meters.
this is SST.
Yeah.
And then this is the integrated depth. So I think, part of it is just these are anomalies, so it gives you a way to compare, like the amount of warming. And then this gives you the integrated depth, which tells you more about, what the temperature was throughout the 250 meters. Yeah, and I mean, so this goes through 2020, but you can see we've more often than not been quite warm. Ben?
We'll go, I don't know.
In 2019, where the fish went that didn't make it past Spring Canyon.
Well, those red dots on that map were the locations of the fish. The locations of the tags.
I guess I was curious if they're like, if they're dying in the channel or if they're giving up and going up and being goodbye.
I don't know. We just know we were dip netting in a lot of these side channels. There were fish there that were noticeably in poor condition.
Yeah.
Yeah, and they were turning red and I know Matt's seen this too, I think. Yeah.
Yeah, that off-channel habitat along the main stem seems to be pretty, I'm guessing it's a critical component, because as water level peaks, there are more fish in those areas, and as it recedes, you will see some fish exit. But certainly some of those off-channel areas along the main stem, there's some fish that aren't going any further.
What about midwinter warming trends? So you have this rainfall in the wintertime, perhaps washing out eggs and also warming the rearing lakes for fry,
Right.
which makes it more of a burden to get through the winter for the fry, for instance.
Right. Yeah, because they found that, yeah, it's a size selective mortality. So if they don't get to a certain probably get to a certain size, then they suffer higher mortality. Yeah. And then the timing of the migration could be, yeah, the smolt migration could be changing too.
Right.
That could affect their survival.
So as these glaciers recede, there's more silt, sand, which has a pretty high iron content in it, and that gets blowing down the river in the wintertime when this bare ground is exposed because of lack of snow, and that would tend to fertilize the ocean from the mouth of the copper and increase survivability of the young fish to get out to sea.
That's right. Yeah. Yeah.
And
When there's enough light for photosynthesis.
All right.
Hey,
Thank you, guys.
thank you for coming.